So what is NAD? So NAD, everybody learned about it in high school biology and then promptly forgot about it. It may have been the reason why people left science or became disinterested, because it is the most boring molecule in biology. It's used for about 500 chemical reactions. It's essential for life. Without NAD, we'd all be dead in about 30 seconds. But, we were forced to learn these chemical pathways. It's not that interesting. Things got really interesting when we realized that the body was sensing the levels of NAD to determine how it was doing and what the environment was like. So we now know when you exercise and when you are hungry, your NAD levels go up. It turns out that as you get older, NAD levels go down. And that's just a correlation. But what we've shown from numerous animal studies, rodent studies, an increasing number of studies in people is that what's going on is the NAD is turning on the body's defenses against aging through a group of enzymes that we worked on and have essentially spent 25 years figuring out. They require NAD as well. So the hormesis mediators, the enzymes that we think provide the health benefits of diet and exercise, are responsible for the longevity that you get when you lead a healthy lifestyle. Now, it's interesting to pause on this because we used to think that just running would make your blood flow better, and being hungry, you lost weight and that's why you lived longer. But actually what I believe, and more people also believe, is that NAD and these other things that raise NAD are working, also resveratrol, are working because they're actively turning on the body's defenses against aging. And it's not a passive effect, it's not a coincidence. It's really just telling your body times are going to be tough, fight against this onslaught. And NAD is one of the best things that I know to turn on these sirtuin defenses.
So an NAD booster.
Yep, how much?
I take a gram of an NAD booster every morning. And it's water soluble, so just take it with a drink, with a coffee, along with the metformin. I also take a gram of resveratrol, which isn't a strict amount. I actually just shake some powder into a yogurt and mix it up, a few spoons of yogurt.
These NAD boosters, and we probably should definitely get into that, but...
Well, they're central too, because as I mentioned, the proteins, many of them, like the sirtuins, are moving around, controlling the epigenome. You want to stabilize that as best you can. Animals like whales and naked mole rats have a very stable epigenome, so this moving around of proteins and epigenomic noise accumulating. If we're exercising, we're taking NAD boosters, we do slow that process down, I believe.
Well, let's talk about what NAD boosters are. So the precursors for NAD, right?
Sure.
We make NAD in our bodies.
Yeah, we do. And so NAD is recycled in the body because there's grams of it. You can't eat that much easily. And there's a cycle, it's called the Salvage Pathway of NAD. And it all starts with nicotinamide, which is a form of niacin, vitamin B3. But you can't just, well, you can, but it's not very effective. Just overdose on vitamin B3 because you need other things to make the big molecule NAD. So NAD is, the reason it's called nicotinamide adenine dinucleotide is that it's got these three main components, and the dinucleotide is related to DNA, right? But that's beside the point. It's a big molecule so that if you give a big molecule to cells, it doesn't get taken up. So we don't feed animals NAD, and we don't just feed them nicotinamide, which is the little N part of NAD, because it's too small in that you need these other parts. So NMN and NR are two molecules, so it stands for nicotinamide mononucleotide, which is essentially the precursor, the immediate precursor to NAD. If you give a cell NMN, it will be taken up by a transporter, which was just discovered by my buddy Shin Amai. We used to work together at MIT, now he's at WashU. A few weeks ago, he wrote about it, I wrote about it, that there's a transporter that sucks up NMN, and the NMN is converted within one step to NAD in the cell. And now it's locked, it's a big molecule, it's locked inside the cell. And that step is carried out by an enzyme, it's got a name, it's called an M.I.T, and that enzyme goes up under stress and calorie restriction. And in yeast, it's the same step. And so we showed years ago that that step of conversion of NMN to NAD, or in yeast, nicotinic acid to NAD, is the critical step for boosting NAD when you're going through your circadian rhythms, when you exercise, when your cells are stressed. And without that step, you don't get the benefits of calorie restriction and your organs start to get old. So what is NR? So NR is fairly popular. A lot of people have heard of it. It stands for nicotinamide riboside. And all it is it's just a smaller version of NMN without a phosphate on there, so there's no phosphorus on it. So if you take NR, your body has to first put on a phosphorus, and then it has to basically link two of them together to make the NAD. So with all that said...
NR gets converted into NMN first, and then into NAD?
Yes. Yes, it has to, yeah. But NR and NMN have both been shown to raise the NAD levels in animals and in humans as well. And there's small nuances about the differences, but they both seem to be effective. Not just in humans, actually, I should say in mice, but in yeast, they work as well, which is nice.
The important point to address is whether nicotinamide riboside or nicotinamide mononucleotide can reach other tissues intact and directly form NAD without going through that NAD recycling pathway that I mentioned earlier called the salvage pathway. The salvage pathway would mean that nicotinamide riboside or nicotinamide mononucleotide were first metabolized into just nicotinamide before forming NAD instead of directly forming NAD. This is an important point because NAD produced from the salvage pathway is subject to feedback inhibition and therefore cannot raise NAD levels in tissues above a certain level. So let's talk about some details. An animal study using isotope tracers allowed NAD made directly from nicotinamide riboside or directly from nicotinamide mononucleotide versus NAD made from nicotinamide via the salvage pathway to be measured. What the study found was that at a low oral dose of around 50 milligrams per kilogram body weight of either nicotinamide riboside or nicotinamide mononucleotide, they produced very low levels of NAD made directly from those precursors, but only in the liver, not in other tissues. Low levels of nicotinamide-derived NAD, on the other hand, were found in the kidneys, and very low levels of nicotinamide-derived NAD were found in the muscles and also in the brain. The human equivalent dose of 50 milligrams per kilogram body weight is roughly 4.07 milligrams per kilogram body weight. So for a 180-pound person, that is approximately 332 milligrams of either nicotinamide riboside or nicotinamide mononucleotide, which is a pretty doable dose, but very little increases in NAD were found, at least in animals, at that dose. A higher oral dose was also done, but only for nicotinamide riboside. A dose of 200 milligrams per kilogram body weight of nicotinamide riboside showed no difference compared to a low dose in terms of making NAD direct from nicotinamide riboside in any other tissues other than the liver. However, more of the NAD derived from the salvage pathway was found in the kidney, muscle, and the brain than at the lower dose. So 200 milligrams per kilogram body weight translates to around a human equivalent dose of 16.3 milligrams per kilogram body weight, which for a 180-pound person is about 1.3 grams, which is pretty high. When nicotinamide riboside or nicotinamide mononucleotide were given intravenously at varying doses, so 50 milligrams per kilogram body weight or 500 milligrams per kilogram body weight, directly produced NAD was found in the liver, kidney, and muscle in a dose-dependent manner. However, the only NAD detected in the brain was that which was salvaged from nicotinamide, suggesting that neither nicotinamide riboside nor nicotinamide mononucleotide crossed the blood-brain barrier. It is noteworthy that head-to-head comparison of identical doses of injected nicotinamide riboside and nicotinamide mononucleotide produced more NAD made directly from nicotinamide riboside in the liver, kidney, and particularly in the muscle tissue compared to nicotinamide mononucleotide. So, what does this all mean, and should we care? What this data from the isotope tracer studies means is that even at very high oral doses, neither nicotinamide riboside or nicotinamide mononucleotide appeared to directly be transported to other tissues other than the liver, at least again at those doses that were measured. However, nicotinamide riboside and nicotinamide mononucleotide were converted into nicotinamide, which was then transported to other tissues, and some of that nicotinamide was then converted into NAD. And at the end of the day, isn't raising cellular NAD levels what is most important anyway? The other animal studies I previously mentioned that showed positive health benefits in tissues like the muscle or the brain were at a very high oral dose of nicotinamide riboside and nicotinamide mononucleotide. In fact, in some cases, they were double the dose, so they were about 400 milligrams per kilogram body weight. So it's possible that nicotinamide riboside and nicotinamide mononucleotide can be transported to other tissues other than the liver at very, very high oral doses, but that's yet to be shown. However, it's also possible that at very, very high doses, the NAD derived from the salvage pathway was high enough to do something beneficial. The isotope data also suggests that if nicotinamide riboside or nicotinamide mononucleotide is administered intravenously, both of those compounds are able to be transported to other tissues and directly form NAD and not be subject to feedback inhibition. This also raises NAD levels in multiple tissues to a much higher concentration than otherwise would be. Of course, IV injection of these NAD boosters is very challenging to translate to humans. You may be wondering why all the messing around with NAD boosters like nicotinamide riboside and nicotinamide mononucleotide in the first place. The reason nicotinamide riboside and nicotinamide mononucleotide are popular is because they can be transported into multiple tissue types, including the liver, kidney, muscle, and heart. The brain is the exception. Neither nicotinamide riboside nor nicotinamide mononucleotide seem to be able to directly cross the blood-brain barrier, but both form nicotinamide, which can be transported into all tissue types, including the brain, where it can then form NAD. So why not just go to the source and take or inject NAD directly? Well, for starters, NAD has poor bioavailability. Animal studies have shown that upon ingestion, orally administered NAD is primarily digested into the precursor nicotinamide, but also to nicotinamide riboside and nicotinamide mononucleotide before being absorbed. While oral bioavailability of NAD is low, the hope is that intravenous NAD infusion may bypass that digestive system. The problem is that no mammalian NAD transporter has been identified, and with the exception of the brain and the heart, extracellular NAD has not been shown to be taken up into tissues. When NAD was injected into the abdominal region of mice, it was able to raise NAD levels in some brain regions. Similarly, mice that were injected with a high dose of NAD had increased levels of NAD in the heart and also protection from cardiac hypertrophy. This suggests that direct injection of NAD at a high dose may be doing something beneficial, at least in the brain and in the heart. I would like to mention that just because no data exists, that does not mean that NAD delivered intravenously cannot raise NAD levels in other tissues other than the brain or the heart. It's possible that extracellular NAD could be metabolized to nicotinamide, and that could be transported to other tissues like muscle and also be converted into NAD. While there is very little preclinical data and zero clinical data on direct IV injection of NAD, it does seem like this may also be a good idea or a good area to explore as a potential way to boost NAD levels in tissues.
What role it plays, in the aging process, it seems as though, at least in some of the studies that I've seen, that mitochondrial function really seems to be important. And yes. I know that you've shown that the sirtuin 3 in the mitochondria itself seems to be really important for the mitochondrial function, keeping your mitochondria young. Yes. And that mitochondria play a very important role in aging as well.
A critical role. Many of the aging pathways that we know, be it the unfolded protein response or mitochondrial biogenesis, all point to efficient mitochondria as one of the key ways to stay young. And one reason is because this is a way, so you can generate energy from glycolysis, which is glucose, carbohydrates, which is we know is linked to aging, but also via mitochondria via a process called oxidative phosphorylation. And that process is not necessarily depending on glucose, but is dependent on an efficient mitochondrial function. So we talked about NAD-dependent enzyme. Many of these enzymes reside in the mitochondria. And we found that for example, the NAD supplementation that is being tested in a variety of aging models requires SIRT3 quite often. This mitochondrial sirtuin, yes.
Nicotinamide riboside does?
Yes. For example, the paper we've published was focused on the age-associated or noise-induced loss of hearing. So if you actually subject mice or humans to very acute hearing, acute noise, they have a dose-dependent loss of hearing. You can protect mice completely from this effect by supplementing with NAD.
Wow.
But with nicotinamide riboside. So if you're going to a rock concert and you want to protect yourself, and this is definitely something
Harley Davidson riders.
Yes. In mice, it actually had an enormous effect, and we found that this effect was dependent on SIRT3. So in SIRT3 knockout mice, the effect was lost.
Do you know if if that was dependent, the SIRT3 in the mitochondria of stem cells, or was it just any cell? Or do you not know?
In the case of hearing loss, it's dependent on some really uniquely sensitive cells in the inner ear. But generally, there's been a sort of an assumption in the field that most of the effect of nicotinamide riboside's protective effect are dependent on SIRT1. I think
Oh, really?
Yes, and in some cases it is. In this particular case, noise-induced hearing loss, it was really clearly SIRT3.
So even the studies that have looked at, for example, I think there's there was a mouse that had some sort of mitochondrial disorder.
Yes.
Even that one was dependent on SIRT1.
No, actually, I did not. In that case, I think the assumption was that it might be helping global mitochondrial function. And so there's a growing number of cases, in, for example, DNA damage associated disease, where you see accelerated aging, where people have been testing the effects of supplementation, because those conditions are associated with increased PARP activity, just like you mentioned. And in those cases, I think it hasn't always been clearly mapped what is the real target of NAD that is dependent on for the beneficial effect.
Do you know if, so NAD levels do decrease with age. Do you know, has it been looked at, like, for example, in animals, like in rodents, when they're fasted, because fasting increases NAD when they're fasting and they're older, does that help rejuvenate the NAD irrespective of their age? Does it?
I don't know the answer to that question. It's a good question. I think, one thing that I remember also about fasting, another critical effect of fasting that is really essential in our thinking is autophagy.
Yes.
And so, there's growing evidence also that autophagy by itself responds to nutrient availability. So NAD levels, acetyl CoA levels. This is a new area that people are starting to work in, and including my labs, trying to understand what is it about acetyl CoA, which is another, think about them, currency exchanger in the cells, intermediary metabolite, that appears to have not only role as a nutrient, but also as a signaling molecule. Acetyl CoA is in the Krebs cycle. It's one of the key intermediary products of the Krebs cycle, but it's also the substrate for a whole family of enzymes called the acetyltransferases, which are the opposite of what we were talking about, the HDACs earlier. So I think there's really a lot of crosstalk between all of these pathways, and we're working very actively now on modulation of signaling via acetyl CoA and these acetyltransferases. And yet another approach is to mimic calorie restriction in the fasting state.
Yeah, I had Dr. Guido Kramer on the podcast.
Yes, exactly.
And he has been studying that as well. It brings up a question I remember I wanted to ask you that was sort of, biology is never just black and white, because I remember him with his work, he was talking about how important decreasing protein acetylation was. So for activating autophagy, which happens during fasting, but also during fasting, you have these beta hydroxybutyrate, which is now was a class 2 inhibitor?
Class 1
inhibitor of the histone deacetylase, which is kind of the opposite, a little.
No, because you have to think so. Beta hydroxybutyrate, it's one step above the sirtuins, and actually the class 1 inhibitors are inhibitors of...
Oh, they're inhibitors of. That makes sense. Okay.
And so it fits perfectly if you go back to the...
So class 1 is an inhibitor of histone deacetylase.
Yes.
That makes sense now.
Yes.
Okay.
So...
Thank you. There was a disconnect in my brain when I was trying to...
You know, because so the whole idea of, if you think about acetyl CoA, and we really draw a graph that of acetyl CoA regulating HATs, histone acetyltransferase, and NAD regulating the sirtuins, and these enzymes quite often say in opposition. Not all of them, but one of the key players in terms of histone acetyltransferase is P300, and that's the one we've worked on, and Guido Kremer as well has worked on. And it makes sense. You get the same beneficial effects by activating a sirtuins, which lowers acetylation, as you do by inhibiting an acetyltransferase, which also lowers acetylation. So the message is that nutrient feeding or low histone acetyltransferase activity or high sirtuin activity all lead to low protein acetylation, which is beneficial.
And doing the ketogenic diet
regulates one step above.
Does also,
Yes. But obviously there's some complexity because there are some histone marks that are depending on one enzyme. So this is an oversimplified model, but so far.